Hello everybody, I'm Matthew Johnson. Today we'll be talking about dissecting complex traits using maze nested association mapping populations. This is what I use in my lab to better understand the genetic architecture of various traits in maze. It is important to understand where the NAM panel came from. It was a collaboration between Ed Buckler, Jim Holland, and Mike McMullen. That's between Cornell and the USDA. It spans a diverse group of maize recombinant inbred lines and captures an estimated 57% of total maize genetic diversity. Here you can see the examples of the NAM founders. These are the diverse genetic maize recombinant inbred lines that were crossed to B73 to make the nested association mapping population. The maize nested association mapping panel was made by taking 25 unique recombinant inbred lines, 26 if you include MO17, and crossing them to B73. B73 being the line that was used to sequence the maize genome. The offspring from those crosses were selfed for about 5 generations until there were 200 recombinant inbred lines for each cross. These recombinant inbred lines total 5,200. To use these 5,200 unique recombinant inbred lines to dissect complex genetic traits, they are all grown in a field or greenhouse conditioned and phenotyped for the specific trait that is trying to be understood. That phenotyping data is analyzed with the genotype data that is publicly available, which contains millions of SNP markers throughout the maize genome. Statistical analyses are able to be used to show which regions contribute to the trait in question. Once the QTLs are identified and the significant ones shown, they can be used in future breeding efforts or whatever other type of research is desired. The nested association mapping panel has already been used to better understand the genetic architecture of flowering time in maize, resistance to southern leaf blight, day length, and leaf architecture. In my research, I'm using the NAM panel to assay salt tolerance. This will hopefully be able to show all of the QTL that are responsible for salt tolerance in maize.